I think. Okay, I've never ever recorded myself on Zoom, so hopefully it works. Okay. You can all hear me, and let's go. So, and I'll be real quick. So, especially for Andy, I apologize, but it's a lot. If something is really something that you want to discuss, stop me or ask me maybe at the end. But we've already done it like so many times that if I go slow now, Anna will feel like I could do this better than him. Why am I here? Okay. Oops, I think I did not share the screen yet. All right. Let's go. Share. Okay. So I'll start by talking about meninges. Meninges, as I all right, let me start by talking about meninges. Meninx is a Greek for membrane or a wrap, a covering. And you do want to know from top, superficial, to bottom. We have 3 layers. So 1, 2, 3, dura, arachnoidea, and pia. But you want to remember it. You want to remember the function of the meninges. They are a wrapping that holds the CNS together. They are a wrapping that holds the brain and the spinal cord together. So it also provides support. And so you want to think of a pad. You want to think of a pad, and you will know that pad means pia, pad means arachnoidea, pad means dura. All right, let's talk about these 3 layers a little bit more. So how do you remember it? Mater, mater, mater. Mater means a mother. Dura means a hard mother in Greek. Soft mother would be your pia. How do you separate the dura and pia? Well, dura is stuck to the calvarium. It's a very hard layer. We'll do it on the next slide. Pia is stuck to the brain. It's a very soft layer. So now you want to separate that hard mother from the soft mother. And what do you throw in there? You throw in spiders in there. Oid means like, arachno means spider. So arachnoidea means spider-like. So you have calvarium, and you have dura. Slapped on it is arachnoidea. Arachnoidea has a cobweb-like structure, and that cobweb-like structure touches pia. Don't forget that this cobweb-like structure is actually your subarachnoid space. Okay, so arachnoidea, the arachnoid membrane, and this space is the subarachnoid membrane. It will come up in a few slides. All right, let's move on. So dura mater is very dense, and it's so dense and so hardy because of this connective tissue that is very irregular. That means that the connective tissue does not run parallel. It runs in all 3 dimensions and provide that extra layer of rigidity. Well, we have 2 layers of dura mater. One of them, as I mentioned, is touching the bones. So bone in Greek means ost, anything to do with ost, ost, bone. Periosteal means what? Around the bone. So the first layer, the one that is touching the bone, is called periosteal layer. It's touching the cranium. The other layer actually touches the other 2 meninges of the pad, of the pia and the arachnoid. So we call them meningeal layers. Okay, so think about it. You have a double layer of dense dura. Next, these dural layers, and we're talking about the meningeal layer, sometimes find each other. They separate from the periosteal layer and find each other and make a fold of dura mater. And there are a total of 4 of them. Look at the number, 2 times 2, 4, all caps. Okay, how do you remember this? Well, there are 4 of them. 2 times 2, please remember. Okay, all right. So these folds can be either vertically oriented or they can be horizontally oriented, either vertical or horizontal. Those that are vertically oriented, like so, sagittal orientation. Okay, both have falx in their name. Falx means sickle. Modern day sickle would be your crescent shaped, okay? Because hardly anyone knows what a sickle is unless you've worked on a farm, okay? So falx. So both of those vertically oriented dural septae, we call them septae for fold, Greek word, a separation. They have falx in their name. One falx separates the left and the right parts of the cerebrum, right? So we think about the brain as a ball. So we think about the right side of the ball and the left side of the ball. And if we cut it into two, we'll have two halves. And so we use the Greek word hemispheres. Hemi means half, sphere is a ball. So the right and the left cerebral hemispheres are separated by the largest fold called largest septum, and we call it falx cerebri. Okay, now the little brain, the cerebellum, also has the right and the left side to it, and those two sides are separated by falx cerebelli. Please note the difference. Cerebri versus cerebelli. Falx is present in both, and once again, falx is a fold of dural, is a dural fold that makes that, that makes that separation, and we call it falx cerebri in cases of vertical orientation. Now, two other separations, two other septae are horizontally oriented. So here is your vertical, here is your horizontal orientation, okay? One of them is called tentorium. And look at the name, tentorium cerebelli. So tentorium means what? It means a tent. So obviously it's a tent above the cerebellum. And what is, if there is a cerebellum here and there is a tent-like structure above it, that means something else is on top, because cerebellum is under the cerebrum. So tentorium cerebelli separates the cerebellum. Where is my, I'm not writing. Okay, cerebellum from the cerebrum, and the two lobes that it separates the cerebellum from would be the occipital lobe, responsible for vision, we'll get there, and temporal lobe, responsible for hearing and memory, we'll get there as well. So once again, cerebellum is separated from the cerebrum by tentorium cerebelli, which is a horizontally oriented dural septum, and the two lobes that actually are separated because of this, from the cerebellum because of this, are the occipital and temporal lobes. One more thing that you need to know, the dural septae do is they form dural venous sinuses. So while making these dura, as a byproduct, you form pipe-like structures all around the brain. I'll show you the animation in a few. So these pipe-like structures are lined on the inside by a layer that prevents blood from clotting. So once again, the pipe-like structures have a lining called endothelial lining. Those are the cells that ensure that blood inside those pipe-like structures does not clot. And because of that, we say so this is going to be venous blood as it drains the parts of the brain. Arterial blood comes to the brain, venous blood goes away from the brain. So these dural venous, what we call sinuses, dural venous sinuses, function as veins, and the way they are formed is that a single layer of dura, like so, for example, comes and meets another layer of dura previously there. So if you just add some kind of a cover, there you go, you will have an empty space. They are empty spaces, and don't forget, on the inside, they're lined by the endothelium. Dura is a lot of connective tissue. If you did not have endothelium on the inside, blood would clot in no time, okay? So, yeah, do not forget that dural septae are projections that actually stabilize the brain quite a bit, right? Because they reach out into the inside of the brain, and brain is much more stable because of that. Let's have a quick look at this. Here are your meninges, dura mater in green, periosteal layer in purple, meningeal layer in yellow. And in there, between those two, that was the dural venous sinus. You can watch those videos a little bit slower now. Now you in red, you see pia. Let me do it again. Oops, sorry, that's a different one. All right, so here you go. You have two layers of dura. Now you're going to see dura periosteal in purple, meningeal in greenish, and in between them, that's your dural venous sinus. That's where blood is flowing. Now in green, you see arachnoidea with all that cobweb-like structure. Inside is the pia, and we'll talk about it right now. All right, so here are the sinuses. Looking at the brain from above, dural venous sinuses. So you see, it's not just the triangular structures, it's actually vein-like structures. So they collect together and drain the blood from the head, eventually to the heart, right? So quite a few of them, too many names, not a 6-week course for sure. All right, so now let's talk about the other two meninges. So dura was a hard mother, right? So we have pia and arachnoidea, and we have put them, we use a term leptomeninges for both of them because they're both thin, lepto meaning thin. So pia mater on the inside, arachnoid mater on the in between. All right, so both of them are very thin. Both of them are very delicate collagen and elastic fibers. And if I tell you elastic, obviously, what do you think? You think, kind of elastic. So we don't think of something hardy. We think more of something that is very supple, that is capable of being very gentle. And therefore, both are very delicate. So the claim to fame of the arachnoid mater, with a name like oid, like spiders, right? Looks like spiders, is obviously that cobweb-like structure that you just saw earlier. Okay, cobweb-like projection. The claim to fame of pia is nothing other than it follows the brain surface contour very, very closely. So if the brain goes down, pia goes down. If the brain goes up, pia goes up. Pia cannot be separated from brain. Pia is stuck to the brain. Dura is stuck to the calvarium. Dura forms those folds. That's the only time it comes away from the calvarium. All right. So please remember that instead of that hard, irregular dural tissue, you have a so-called areolar connective tissue that is very flexible. Hey, it's elastic, right? And it's got some cells. Sorry, it's got some cells. It's got some fibers. It does provide some support, but it's pretty much a support in the name only. It's important, but you're not talking about the level of the support like bone or the level of support like dura earlier. Now, brain is mortally scared of blood. Okay, brain is mortally scared of blood because blood is toxic to neurons as is. So we want to filter it. We want to allow some parts of blood to actually get to the neurons. Whoops. Some parts of blood to actually have an access to neurons, but not all of them. Okay, so we have a structure made up of 3 layers, 1, 2, 3, that we call blood-brain barrier, abbreviated BBB. You will see both of it. You will see actually BBB used more commonly. All right, so let's start by talking about number 3 first. Here are your endothelial cells. You still are learning anatomy, so let me tell you what endothelial cells do. I've already used this term earlier. I told you endothelial cells actually line the dural sinuses on the inside so that blood does not clot. And that's exactly the function of the endothelial cells. What they do is actually they line all the blood vessels so that blood doesn't clot. Okay, every single blood vessel we have. And so endothelial cells of the capillaries, and that would be your very smallest blood vessels that we have in the body, are very, very tightly bound in the brain. Very, very tightly. You can't separate them easily. Nothing can go in between. And we call that tight bonding, we call it tight junctions. Okay, so the endothelial cells of the capillaries in the brain are impermeable. They have very tight junctions. So let's call it this pipe. The blood vessels are essentially pipes that carry blood, right? And so let's call this the blood vessel, the capillary. And so what you should understand is that on the inside, okay, that's your endothelium, that's your endothelium, okay? And so endothelium needs to sit on something, and it does sit on something. That's your basement membranes, just a structure that you can sit on, and it's only it surrounds the whole endothelial cell layer. Why so? Because how else are you going to sit? You can't just sit like on a side, right? So what's really interesting is that to get to a neuron, that's not sufficient. So we now have endothelial layer protection from blood. We now have a basement membrane. That's not enough. The brain needs one more layer of protection from blood, and that's the layer created by astrocytes. Cytes means cells. Astros is given to those cells because they do look like stars a little bit. I'll show you a sketch on the next slide. These processes and processes, whenever you see processes, it just means something coming out, okay? Are often look like feed. We call them astrocytic feed processes or something, or food processes of astrocytes, okay? So this is how the blood-brain barrier is made. Now, do know that there are a couple of places in the brain, about 7, where this blood-brain barrier is reduced or missing altogether. And that's okay. We do need that too. In fact, we take advantage of it because that's how medications going to brain can actually get to where they're going, okay? So the blood-brain barrier is reduced or missing. Remember these 3 structures. Hypothalamus, that's something we'll be discussing. That's a part of the diencephalon. Pineal gland, hey, that's epithalamus. It's the biggest part of the epithalamus, obviously diencephalon as well. And the choroid plexus, and we'll go over choroid plexus in the next 1 or 2 slides. Choroid plexus is the factory within the cell that actually makes brain water, the cerebrospinal fluid, brain water, cerebrospinal fluid. Choroid plexus. Please remember the name. It's not very easy to remember, but I'll show you the picture of it. Okay, so let's go over the picture now. So here is your astrocytic cell body. Look at all those processes, and they do form foot-like structures. So scientists like analogies, and so they said, hey, this looks like a foot planted on the blood vessels basement membrane. So let's look at all the 3 layers. Here is your endothelium on the inside. It's sitting on the layer called the basement membrane. That's the capillary, and the capillaries are separated from the neurons, right? There are no neurons shown here. They are separated by this astrocyte. Whoops, sorry, once again. Astrocyte, okay? They are separated by the astrocyte. So brain would be somewhere here. Oh, sorry, the neuron would be somewhere here, okay? So you need to pass all of these 3 layers to actually get to the neuron. Okay, now, and so we call this the blood-brain barrier. Notice, these are very, very tightly bonded together, tight junctions, okay? And let's now talk about a modification of the blood-brain barrier. I told you there are 3 structures that you need to know for sure: hypothalamus, epithalamus, that's your pineal gland, and the choroid plexus. Now, let's go over specifically the choroid plexus. Does the choroid plexus have an endothelium? It sure does. There it is. But what's really interesting is that endothelium is not very tightly bonded. Look at this. There are spaces here. And so material in the blood can actually squeeze through. And not only that, there is something else. Instead of astrocytes, we don't have astrocytes in the choroid plexus. Look at this. These are astrocytic repeat processes. They're very, very tightly bonded to each other. Here we have another cell type, and we call them ependymal cells. Okay, you don't need to know it, but the ependymal cells are among not for chapter 13. You do need to know it in a few classes down the road. But these endothelial cells wrap around the basement membrane, and what's really interesting is that they can actually produce water. How do they produce water? Well, blood is made up of red blood cells. Most of the cell, like red blood cell is the predominant cell in the blood. That's why our blood is red, right? We have a few white blood cells. We have a few white blood cells. We have a few platelets. Those are the three cell types. We have lymphocytes, okay, which are also white blood cells, okay? So those are the cells, okay? What else do we have? We have cells and a lot of water, in which we have a lot of protein, like albumin, antibodies, etc. They're all swimming in blood surrounding the cells, okay? So guess what? Out of that, so about 45% of blood is cells, and about 55% of it is plasma. Plasma is water plus all those proteins, etc., etc. So what's interesting is that out of that 55%, 90% is water, just your normal water. And so what these cells, the ependymal cells, can do, they can take that water and separate it with a little bit of ions like sodium, like potassium, etc., etc., and a few of the necessary things that are important for the brain. Okay, so the ependymal cells, their marker would be that they have all of those structures on the outside. Okay, but this trio, the endothelial cells, the basement membrane, and the ependymal cells, that's your choroid plexus. Okay, that's the special portion that evades our commonsensical understanding of the blood-brain barrier. In what sense? Hey, it's not a real barrier. It's actually almost permeable. No tight junctions, and makes water. Ependymal cells make CSF. All right, so CSF is made in the choroid plexus. We have choroid plexus in all the ventricles. How many ventricles are there? There are 4 ventricles. And how do you remember that? Well, the last one is the 4th one, so it's easy to know. They're all numbered, okay? And the 4th ventricle is numbered 4, so it's called 4th ventricle. 3rd ventricle is numbered 3, so it's called 3rd ventricle. And ventricles 1 and 2, they're the only exception. They're the only exception, but they're both on the kind of the side of the brain, okay? So we call them lateral ventricles. I'll show you the picture in a second. Lateral ventricles, okay? These are the brain ventricles located in the cerebrum, okay? The 3rd ventricle is located in the diencephalon. You've already studied this for a few times already, so I'll just remind you, diencephalon means what? Thalamus. The top of the thalamus is epithalamus, the bottom is called hypothalamus. And the 4th ventricle is located between the pons and the cerebellum. We'll go over that as well. And there is an interesting partition between the lateral ventricles when they come together. And that's called septum. That is kind of translucent, and we call it septum pellucidum. Don't forget that septum means a separation, a wall. We had dural septae earlier, right? So let's look at the picture of the ventricles. Here is your 3rd ventricle, about this much. This is your 4th ventricle. There it is. And these are your lateral ventricles. They look like, on this picture, they look like a C-like structure, letter C-like structures. If you look at them laterally, you will see that they actually form what we call horns. Okay, so it's within these ventricles that the choroid plexus produces CSF. Okay, it's within these ventricles. And by the way, you see here, these two come together, and that's where we have this septum pellucidum. All right, next topic. What happens to all that water? Because the brain makes about a venti cup of coffee, 600 mils of water in one day. Look at the number. It's not 600. It's 125 to 150. This is the number that you need to remember. So 600 mils is made per day, but hey, it keeps on flowing downhill. And at any given time, the total volume within the brain is about this much, 125 to 150 mils. That's your short cup of coffee, if you like coffee, okay? So cerebrospinal fluid, what is the function of it? Well, obviously protection. So if you were to jump off a cliff and you were to land in water, you would not be hurt especially if you know diving. But the chances of being hurt when hitting water are much less so than if you were hitting a bone, which is your alternative for a brain, right? We have a calvarium. So the brain needs that cushioning, needs that fluid cushioning for protection and for buoyancy, okay? Kind of reduces the weight, it kind of makes the brain flow, and we'll see later on that brain has so much fat that floating is pretty much a given, okay? It also provides a very stable chemical environment. So it provides this chemical stability by clearing waste, by replenishing things that are lost. But think of clearing waste. That's very important because we're talking about a brain which is a collection of cells, right? So obviously cells produce their own trash, shall we say, right? That trash is constantly taken away from the area by flowing CSF, just like a river would take some trash on its banks, okay? Total volume, once again, your short cup of coffee, 125 to 150 mils. All right, so how does this river flow? How does this CSF flow? Well, we do know that it flows from top to bottom. Let's look at it from top. This is the top of the brain to bottom. So obviously the bottom would be your fourth ventricle. Let's look at it. Here's your fourth ventricle. Okay, fine. And the top would be your lateral ventricles. The third one is in between, and it is correct. Lateral ventricles flow from lateral ventricles where CSF is made in the choroid plexus. The CSF flows to the third ventricle, so more CSF is made. The ventricle is smaller, so obviously less CSF is made, right? And from there, it flows down to the fourth ventricle where some more CSF is added. But as you know, we're all flowing downhill here, okay? Now watch this. This is the structure that I kind of alluded to earlier. From there, from the fourth ventricle, CSF flows out into the subarachnoid space. And I told you, dura has a layer of arachnoidea attached to it at the bottom. Fine. What happens to that arachnoidea? It has cobweb-like projections that touch the pia. And look at this. All of that cobweb-like projection creates a space in between that arachnoid membrane on the top and pia on the bottom. So I said the membrane on the top. So arachnoid is on the top and that space is below it. And that's why we call it the space that is below subarachnoid space. We'll see that again. And from that subarachnoid space, the CSF flows back to blood, to the dural venous sinuses. So let's review it again. at least 2 times, okay? Let's review it again. Let's analyze it, okay? So you have lateral ventricles to third ventricles to fourth ventricles to the subarachnoid space to dural sinuses. So don't forget that the lateral ventricles take arterial blood from the arteries and make CSF, right? Blood from the, so CSF is made from blood. And then it goes down the route back to the blood. Now this is venous blood, okay? Now this is venous blood. So from CSF is made from the arterial blood and is flows to the venous blood vessels, dural venous sinuses. So basically you have like a river that is made, because of all those tributaries, all that, rain, etc., right? And where does all that water flow? Into the ocean, into the, into the sea. So dural venous sinuses, think of them as that ocean, that sea. And don't forget that the lateral ventricles, third and the fourth ventricles, make CSF from blood. It's very important to understand it, that choroid plexus takes the blood and filters it and makes CSF, all right? Now, you also do need to know more detail about CSF flow. And those details come better if I show you the pictures, okay? So we have a lateral ventricle. You somehow need to get here to the third ventricle, okay, in the diencephalon. So obviously there should be a hole and there should be a pipe-like structure connecting the two. Number one, from the third ventricle, you need to get to the fourth ventricle, right? So obviously here too, you need a pipe-like structure, this time a little bit longer, by the way, that connects the two. And they both have names. Now, what did I tell you? From the fourth ventricle, CSF has to leave and enter the space around the brain, the subarachnoid space, because we said that pia is stuck to the brain, and on top of that pia, we have arachnoidea, and that's the space that is called under that arachnoidea, right? The subarachnoid space. So from here, you want to flow and cushion the brain all around it. This is how this happens. Whoops, sorry. This is how this happens. We have an between ventricular foramen. Foramen means hole. That connects the lateral ventricles. Ventricles means what? A belly-like structure. We have a ventricle in the heart, right? A belly-like structure. Like if you were to take a little balloon, blow it up, it would have that belly-like structure, right? So lateral ventricles have an inside that is a cavity, like a hollow space. So lateral ventricles, hollow cavities in the brain. From those cavities, water CSF flows to the third ventricle via a hole that connects the third ventricle and the lateral ventricles. And that hole, Greek for hole would be foramen, is called the foramen between the two ventricles. Ventricle between the two ventricles. The interventricular foramen. Sometimes they'll call it interventricular foramen of Monro to honor the discoverer, okay? So now, how do you get from the third ventricle to the fourth ventricle? Similarly, you have a water pipe. Duct means what? A pipe-like structure. Aqueduct means what? For water. Aqueduct, that connects the cerebral third ventricle to the fourth ventricle. Sometimes we call this cerebral aqueduct of Sylvius to honor the discoverer, okay? And this way the water gets to the fourth ventricle. From the fourth ventricle, there are actually two different types of holes, foramina, two different names rather, total of 3 holes. One is the lateral foramen and one is the median. And I'll remind you that median means what? When you're driving on the road, median is that separation that separates you going one way from the traffic going the opposite way, the median, and this is exactly what it is. The fourth ventricle has one hole, one foramen, smack in the middle. It also has a name, foramen of Magendie, and there are also two holes to the sides, on the sides, and these two are to the sides, and so we call them lateral. Okay, so lateral foramina. And CSF exits the fourth ventricles via these three holes, and then goes into the subarachnoid space, from where a membrane projection of the arachnoid essentially enters the dural venous sinus and kind of allows CSF to just flow into the dural venous sinus. Let's look at the picture again. So here is your lateral, here are your lateral ventricles. The connection between the two is called the interventricular foramen. Here is your third ventricle now. The connection between the third and the fourth is called the cerebral aqueduct. Here's your fourth ventricle. From here, we need to get out. One hole is in the middle, the median foramen, and two are on the sides, and we call them the lateral foramina. From here, CSF enters the subarachnoid space. Here is your animation of that hole so that you understand. Here's the dura. Dura has been stripped. Now you're looking at the sagittal section with a few, but look at those horns. Those are the lateral ventricles. Those are the parts of the lateral ventricles. And look at that connection between the two, right? So now that's how the whole cavity looks like. It's connected with holes like that. That's your hole. That's your hole. Okay. And now you're in the fourth ventricles, and from there you're going to get out, and you're going to now enter the subarachnoid space. Let me show it to you again. Let me show it to you. Here we have CSF being made in the lateral ventricles. Now it's going to enter the interventricular foramen, the third ventricle, from where it goes via the cerebral aqueduct into the fourth ventricle. Now it spills out via the median and the lateral foramen into the subarachnoid space. And from there, from that subarachnoid space, look where it's going to go. It's going to go, that's your subarachnoid space. You see that cobweb-like projection. Guess what? It's going to enter the dural venous sinuses. And those dural venous sinuses are going to take all of that CSF away. And areas which project into the dural venous sinuses, the parts of the arachnoid that project into the dural venous sinuses are called arachnoid granulations. You can see them somewhat here like projections. I need to magnify it slightly. All right, so this is not really, this is just for clarification. Look at this. This is a transverse section of the brain, right? Cut like so. And we can see the lateral ventricles here. Guess what? That's your septum pellucidum. That's where the ventricle, that's where the lateral ventricles come together, right? So this is a normal, healthy person. Look at the amount of water. We said a total CSF here in the lateral ventricles and the third ventricle and the fourth ventricle, the total amount is about, and the subarachnoid space and the spinal cord. Let me bring it home again, because we're talking about brain. Spinal cord is also wrapped by the same pad, pia, arachnoidea, and dura. Spinal cord and all of CNS, that will be your brain and the spinal cord. So what you're seeing here is the brain water in the ventricles, right? The black H-like structure. Now, how about this? This looks like a huge butterfly, and that's not a good sign. That means that the brain water can no longer exit from the brain. It means that it's stuck in there, and you know, the amount of water is collecting, and look how much water we have. I mean, What do you think is the patient feeling? A headache, that's the least of the problems. All of these neurons are squashed together. Look at the space that the brain, you can't really expand too much, right? Because we have that calvarium that limits the space. And this is hydrocephalus. Hydrocephalus, a problem where there's too much water in the head, in the brain, okay? These are various sections through the brain. This would be your lateral ventricles. Once again, this is third and this is the fourth ventricle, right? And the connections between them. Here we're looking at the brain from the left side. Now we've cut the brain. It's called a sagittal section. We're looking at it from the middle. We're looking at the middle part of the brain. Sorry. And this is an added coloration because blue is for the frontal lobe, pinkish color is for the parietal lobe, and the orange coloration is for the occipital lobe. Similarly, here you have different colors representing the lobe. You know, we have 4 lobes here. You can also see the temporal lobe here. We cannot see the insular lobe. Oftentimes you will never, if you read up on your literature, you may never see the insular lobe as the 5th lobe, because insular lobe, you know, some books list it as a 5th lobe, others say there are only 4 lobes. But the reason I'm showing you this is because I want you to know 2 things. Number 1, it's the frontal lobe, is the parietal lobe, this is the temporal lobe, this is your cerebellum. Same thing here, this is your cerebellum. Remember the dural folds, they go all the way down there, the dural septi, that's your horizontal fold. Guess what? Your, sorry, that's your vertical fold, the falx cerebri, and this is your horizontal fold, that's your tentorium cerebelli. So tentorium cerebelli separates the cerebrum, the occipital, and the parietal lobe. Parietal because on the inside, okay? So a little bit, you can't really see it here very well, but tentorium cerebelli is here, the horizontal fold of the dura. And so the second thing that I want you to look at is the base of the brain. You can see the eyes here, and look at all those projections. Look at all those projections. These are nerves. These are the nerves that brain makes, and these nerves do not stay in the brain. They actually get out of the brain through the floor of the brain cavity, so through the bones, and run to the parts of the brain that they're parts of the body that they're responsible to rule, okay, essentially. All right, now this is a little bit of a better picture. You can now see all of these nerves in yellow. So this is, for example, the cranial nerve number 1, this is 2, and so on. This is 3. So what you can see is as we go down, nerves, the name of the nerve starts with 1 here and 12 here. That's how you remember. We call them C1, 2, 3, 4, up to 12, based on how close they are to the, let me show you, to the tip, to the frontal part of to the most anterior part of the brain, okay? That's the, so if I were to look from here, 1 would be here, and so on, okay? Down the road, and 12 would be somewhere here, okay, in the medulla. All right, so now we're looking at the base. Again, this is how the real brain would look like. You can see all those little hairy structures. Those are the cobweb-like structures. They're very hard to get rid of. That's your subarachnoid space. That's where CSF cushions the brain, provides that buoyancy, provides that protection, okay? So this is your spinal cord. This is your medulla. Notice this like structure, this bridge, the Greeks called it the bridge-like structure, not very prominent here. We call that the pons, okay? All right, I kind of worked on this mnemonic, kind of to remember all the names, all the cranial nerve names. Not very easy to remember, I'll tell you honestly, something that comes with time, okay? You just have to review, that's pretty much it. But what I did here is that I actually used, computer help to come up with a mnemonic that includes the name, at least the first letters of the name in the mnemonic, okay? Because most of the mnemonics use just like one letter, and I thought that's insufficient. I mean, once again, after a while, you don't need any mnemonics like this, but it's a good starter. Okay, so for example, optician for optic nerve, ocular for oculomotor, tron for trochlear, trio for trigeminal, abduction for abducens, faces for facial, and so on. So just if you like this mnemonic, use it. If you don't like it, find a mnemonic that you like. Mnemonics are not mandatory to learn. You don't want to sit there memorizing a mnemonic that doesn't stick to your brain. Okay, you want to, if you like it, use it, for God's sake. If you don't shy away from dropping it and say, I don't like quite a few mnemonics, for example. And down the road, if you're studying other classes, you will see that many people come up with very fancy sounding mnemonics where you may not like them. And sometimes learning mnemonics and too many mnemonics also, they make zero sense. Okay, so just do what is best for you. But this mnemonic has been around, like every single medical student knows it. This is a good one. This, for some reason, sticks to most people I teach, also definitely my favorite. And so you don't need to remember which nerve does what, in the sense, is it a motor nerve? Is that a nerve that goes to a muscle and says, do that motion? Or a sensory? Sensory means what? Feelings. Is it cold? Is it hot? Is it where in space is my arm? My arm is raised right now. My brain knows it because that's the sensation. That's the sensation. You've already studied all of this, or I'll just remind you, all that sensation goes to the parietal lobe. And we'll look at the particular gyrus, the precentral and the postcentral gyrus in a few. But once again, some say marry money, but my brother says big brains matter most, very different variations thereof. But this is a good one that basically tells you that if it's number three, marry, it's an M. An M can only be motor. Money can only be motor. So for example, if I asked you, cranial nerve number 6, does it have sensory fibers or does it have motor fibers? You say to yourself, 1, 2, 3, 4, 5, 6. Some say marry money, but my brother, so number 6, my, right? Motor. There you go. So this is the way you can answer questions. Okay. So I have never ever spent a second memorizing whether a nerve is sensory or motor or both, because this mnemonic does what it's supposed to do. Let's look at just a few of the nerves. Olfactory nerve. Olfaction means smell. Olfaction, olfactory nerve, cranial nerve number 1. How do you remember it? Your nose. If you look from front, right? If you're talking to someone, their nose looks like a number 1. And so olfactory nerve is the cranial nerve number 1. It's the front most anterior cranial nerve. It's toward the nose. Don't forget that we do use the term rostral. Rostral means nostril. Rostrum is the beak of the bird. So we do use this term, anterior or rostral, toward the nose. It's the most rostral, most anterior cranial nerve. It's got its own claim to fame. See that plate through which it has to pass, the bony plate, has lots of holes. And a part of it has even more holes. So we call that bone, the ethmoid bone with a cribriform plate. Both of these, cribriform and ethmoid, mean sieve-like. So there are a lot of holes, and 2,000 to 3,000 of these projections, the cranial nerves, enter the nasopharynx. So inside the nose, enter the nasopharynx, and nasopharynx is obviously where you can, what allows you to smell. Okay. All right. So nasopharynx, cranial nerve number 3, 4, and 6 are responsible for motion of the eye. Cranial nerve number 3, 4, and 6 are responsible for motion of the eye. Don't forget, you want to start with 6. That way you will get rid of it. Okay. Abduction, abducens. Abduction takes away. Abduction takes away. Someone abducted something, abducted a baby. Abduction, hopefully never. Okay. So moves the eye away from the center laterally. And don't forget, some say marry money, blah, blah, blah. Only motor fibers, only motor fibers. So cranial nerve number 3, oculomotor. What does it do? It's a motor nerve, first of all, it's only motor. And what it does, it's a 3. Number 3, so it moves your eye up, moves your eye down, and moves your eye to the inside, toward the nose, up, down, and in. Up, down, and in. There is another nerve, another muscle that is called inferior oblique that has a little bit of a more complicated function. It rotates the eye, moves it, raises it, moves it a little bit toward other sides. Never mind this. Just remember the main function of the oculomotor nerve that is most obvious are those 3 motions, 3 movements, up, down, and in. Cranial nerve number 4 is called trochlear nerve. Trochlear nerve moves the eye, trochlear nerve moves the eye down and in. Down and in, obviously, motor fibers only. And it's called trochlear because if you look at the muscle, that goes to the eyeball, what it does is that it has to pass that pulley-like structure, and that's the reason we call it trochlear nerve. And so if you were to pull this muscle this way, basically eye looks down and in toward the nose, good for going down the stairs, and we call it trochlear nerve. All right, so this is an example when the right oculomotor nerve does not work. Look at this. The left oculomotor nerve works. It's supposed to go either up, or down, or in. The right abducens nerve, cranial nerve number 6, works. So 6 here, 3 here. They are all okay. But look at this. 6 here works. Abducens works. And the oculomotor does not work. You can't bring the eye on the inside. So cranial nerve, so that would be the motion of the eye. That's what we want to focus on here. For example, cranial nerve number 3 is not working well. Cranial nerve number 7 is damaged if someone gets a severe injury to the cheek, or, for example, an inflammatory condition due to a bacterium called a Borrelia burgdorferi spread by a tick in the northeastern areas, and that would be your Bell's palsy. Bell's palsy. Bell's palsy presents with a target-like rash, and followed by damage to the, it's followed by inability to actually move the facial muscle. So, for example, patient has a drooping mouth. Eyelids don't close, obviously, because you need the muscles to close to shut your eyes, right? So cranial nerve 7 controls all facial muscles. There is one exception, but not important at this stage, okay? So what else does it do? It has a few other functions. For your level, you do need to only know that it's also responsible for producing saliva. Some glands are controlled by the cranial nerve 7, innervated by cranial nerve 7. And so saliva, salivary glands, but also tears, lacrimal glands. So when a person has Bell's palsy, it's like a double whammy. The person can't make tears. And not only that, the patient can't shut the eye closed. So that's pretty dangerous. Okay? So remember, goes to the muscles, goes to the glands. So what is this? Okay, I need to get my Kaspersky renewed. I'm forgetting all the time. All right. So paralysis of facial muscles, dry eyes, and dry mouth. That's your Bell's palsy. That's your severe damage to the nerve. If the area of the cheek where it leaves the bone from is hurt, severe damage. Okay, not your slap or something. Okay, so don't forget eyelid droop, sagging at the corners of the mouth. That's not a trivial thing. It takes a few weeks to come back to normal in case of a Bell's palsy, and the patient may have problems, retaining food in the mouth. And what's really dangerous is the eye. The eye may dry out. So use some kind of a lubrication. All right, cranial nerve 10, vagus. So much you can talk about it. Just to summarize, it works on the muscles and the glands, similar to cranial nerve number 7. We had muscles and glands, right? But this one is a little bit different because it goes down all the way from the head to the abdominal organs and the heart and the lungs. So trachea and larynx are obviously, trachea are the windpipes which lead to the lungs. You'll learn it in your other anatomy, anatomy part 2. The larynx is the voice box. Okay, so cranial nerve number 10, the vagus, works on all of them. It is important for speech, the voice box. It is important in your breathing. It's especially important for the heart because it reduces the heart rate. And it's also very important for digestion. And you can actually see it here that the nerve starts the base of the skull, goes all the way down to the intestines. I mean, this is incredible how humans are built. All right, once again, these are the parts of the brain. We're now going to be talking about lobes. Okay, we're going to be talking about lobes. Here is the video overview of the lobes, the video overview that shows your sulci, the lobes. Please watch it at a desired speed. Just remember, cerebellum is in red, for example, then the parts of the brain, diencephalon, which is made up of thalamus, epithalamus, hypothalamus. Now, it will also show you the brainstem, which is the midbrain, pons, and the medulla. Midbrain, pons, and the medulla. We're going to go over all of this. This is just a quick overview so that you've seen it once. Okay, we will start with the development of brain. All right, so in brain development, you need to understand that the portion of the brain that is responsible for actually forming the neurological structure is one of the 3 layers of the embryo called ectoderm. Ecto means on the outside, derm means skin, right? So there is like a plate-like structure that forms that will eventually fold and form a tube, and this tube will further develop into the brain as we know it. Let's look at the details. Once again, rostral means anterior, nostril. Rostral is closer to the nostril, and so anterior or rostral hole of that tube is called the anterior or rostral neural pore. The neural tubes opening nearest to the future head is this pore. I'll show it to you on the coming slides in a few. And this, don't forget that the cerebral cortex consists of the gray matter, will be coming up on the slides, And this gray matter contains the neuron cell bodies. Here is your cell body. Do not confuse that. Axons are the white matter. We'll go over that in a few. Brain lobes are named after the bones that overlie them, protect them, and are a part of the cerebrum, not cerebellum, right? And so brain sulci, brain gyri are ups and downs on the surface of the brain. Sulci, for example, are depressions between the folds, okay? Depressions, okay? Don't forget that pia mater actually follows those sulci, follows those gyri, okay? And the space above pia is called the subarachnoid space. That's where CSF flows out from the fourth ventricle via the median foramen of Magendie and the lateral foramen of Luschka. All right, so now I want to go over the brain development. I think this is also the topic that is a little bit harder to understand. So take a little break. If you're watching this video again, and you want to take a little break, pause here and finish it after a little pause, because otherwise this. I mean, for you probably not so much because you've already seen it so often, right? So all right. So primary brain vesicles. So when the brain is first developing from that tube, and I'll show you the pictures in a few, there are three vesicles, three little balloon-like structures that are separated from each other in ways, and we call the brain inside in the head. The Greeks call the brain inside the head encephalon. So the brain portion that is so the brain would be your encephalon, right? So the brain portion that is frontmost, we call it the prosencephalon, the forebrain. The one that is in the middle, it's called mesencephalon. And rhombencephalon, rhomb actually means diamond shaped, okay? Like diamond shaped, but in English we call it hind brain, okay? Rhombencephalon. Now, these three primary vesicles give rise to 5 secondary vesicles. Please remember that mesencephalon gives rise to itself, the mesencephalon only. Nothing happens here, right? But prosencephalon divides into 2 portions. One is called the telencephalon, the other one is diencephalon. And rhombencephalon, the hind brain divides into 2 more structures. Those structures are metencephalon and myelencephalon, and these metencephalon will further divide, and we'll see how it divides, just to prevent from inundating the slide. All right, so just to review, this is a cell body. This is the axon. Axon has a fatty wrap around it. Fatty, why so? This is your cell membrane. So glia cells that wrap around the axons. Axons are projections of nerves that go to some place to talk to other neurons, that they're sent from that cell body to talk to other neurons, to talk to other muscles, right? So they're length, a little bit longer projections. The longest projection of the cell body is called an axon, and those axons usually have a wrapping, sometimes a little bit of a wrap, sometimes more, and sometimes quite a lot of that. That wrap is nothing but a membrane phospholipid bilayer twice wrapped around it. So this is how it happens. This is your glial cells that wraps around the axon. For example, a Schwann cell or an oligodendrocyte, which we'll study in a few other classes. Okay, so it keeps on wrapping, and basically has double membranes wrapped around the axon 200 times or so, or even more. And so this is a very fat-rich area. So this is The myelin sheath is a very fat-rich area, and I want to remind you that you want to remember it by thinking of a steak. The fatty areas of the steak are white, and so the white matter are the myelinated axons within the brain or the spinal cord, white matter. And the gray matter are nothing but your cell bodies. So as I mentioned earlier, we have that 3-layer plate that forms a tube-like structure. That formation starts in the middle. So it kind of like forms that loop, starts making that loop, almost completely mated, completely mated. That's your loop. It starts around here. This is your completely closed loop, right? The tube. And it starts around here in the middle, and it kind of zips toward the upper side and zips toward the lower side. We said that the upper side is called the cranial neuropore, or anterior neuropore. Pore means a hole, so it's the topmost hole, okay? And this hole will eventually close and form a structure like so with 3 vesicles, 1, 2, 3. Before that, let me remind you that the lobes of the brain are so called because of the bony structures above them, which bear exactly the same name: frontal, parietal, temporal, and occipital lobes. The 5th lobe, the insula, you can see it here inside there, or inside here. You can actually see it on the coronal section. So to see that, you have to push the temporal lobe away from the overlapping frontal and parietal lobes. You kind of have to separate them. Then you're going to see that insula, this is what the brain development further looks like. You have 3 vesicles. Mesencephalon still becomes a mesencephalon, still is called the midbrain, and so on. The forebrain divides into two secondary vesicles. One is called telencephalon, the other one is called diencephalon. And the hindbrain, the ramencephalon, has 2 more portions to it. One is called metencephalon, the other one is called myelencephalon. And here you can see that metencephalon further divides into pons and cerebellum. Myelencephalon forms the medulla. All right, so during this development, the brain starts off as a smooth mass, forms a somewhat more rugged structure, and so on and so on. More and more gyri are seen, and this is our adult brain. All right, now brain ventricles, once again, are seen here. Don't forget them. Brain ventricles, please remember, this is your aqueduct connecting the 3rd and the 4th ventricle. The interventricular foramina of Monro are there. Lateral, median foramina, and the space surrounding the brain, also on the bottom, everywhere. That's your subarachnoid space. That's how your brain water flows, right? CSF. All right, let's talk about the portions, about the parts of the telencephalon called cerebral cortex. Once again, the prosencephalon is the primary vesicle that forms telencephalon and diencephalon. So now we're going to talk about the cerebral hemispheres, and what we're going to address here is the cerebral cortex. Cerebral cortex, that's something that we see on the brain when we think about the brain, the first part of the brain, and those would be your lobes that you've already seen on the pictures here. We have frontal, parietal, we have temporal and occipital lobes. Please remember that the occipital lobe is important for vision. That's an easy one. Temporal lobe, obviously, for hearing. Don't forget, it's under the lateral sulcus. So lateral sulcus, this is your lateral sulcus, okay? shown better here, the lateral sulcus, that opening. All right, so lateral sulcus, which we call Sylvian fissure as well. Parietal lobe is right behind a central sulcus. Central sulcus is seen here. This is your central sulcus. It separates the frontal and the parietal lobes. All right, once again, parietal lobe immediately posterior to the central sulcus. Just remember that name. And parietal means what? Sensation, sensation. So the frontal lobe does what? Motor activity. For example, talking. I'm talking to you right now. That's a lot of work for the frontal lobe. And also prefrontal lobe, mostly decision making. You're planning things, you're solving things, planning things and solving things. Insula Island is the seat of taste. We have information about taste that's processed in the fifth lobe, and that's your insular cortex, or primary gustatory for taste cortex. And I showed you how you find it. Once again, this is the frontal lobe. This is the temporal lobe. This is the parietal lobe. This is the occipital. This is the temporal. And insular lobe is between the two. You have to push the temporal away from the parietal and the frontal lobes at the Sylvian fissure. Okay, so Sylvian fissure, lateral sulcus, central sulcus. All right, so the brain is obviously a seat of the conscious thought processes and origin of all the complex functions, but it's not the same throughout. So the cerebral lateralization exists. That means the left side of the brain does one thing, the right side does something else. And there is a division of functions. For example, we know that the right side is responsible, for example, for facial recognition. How do you remember that? Well, you got to see the face, that's your visual recognition, and you want to see like the parts of the face, the nose, the eyes, the ears. They're different on different people. So you kind of think about it as spatial variations, right? So facial recognition is a good example of that. Visual imagery, spatial abilities, but also music. So that's your right hemisphere, right? On the left side, that's where speech is actually made. So mostly responsible for speech and also more like analytical reasoning versus music. Analytical reasoning such as categorization, symbolization, etc. Okay, there's also one more thing on the left that is called the frontal eye field. That area is called frontal eye field, and that area is really responsible for moving the eyes as you want them. For example, if you want to read a text, you can't read the text if this frontal eye field is damaged, even though you will have a normal understanding, because job of the brain is so well separated. One part of the brain, for example, just moves the eyes. Whoever said you can't understand, of course you can't, but depends on which area is damaged, right? So on the brain, we have so the brain represents our whole body, and how much of the cortex relates to each part of the body can be seen. So a certain amount of cortex is dedicated to moving the body, and a certain amount of cortex is dedicated to feeling what's the information that's coming to the body, right? So the feelings are obviously found in the parietal lobe, and so the post-central gyrus. Just remember that the feet, for example, on the post-central gyrus are actually located medially in the longitudinal fissure. Let's look at the picture. So primary motor cortex, primary somatosensory cortex. Primary somatosensory cortex is your post-central gyrus, so-called because of that central sulcus, okay? So feet are shown here, and they're located medially. This is your median. They're located on the toward the inside. They're called located medial. Okay. Look at the lips. Lips have such a big dedicated area. Look at the motion of the tongue. Look at the motion of the lips. Same thing here. Look at the motion of the hands. Look at the sensation to the hands. So much less is dedicated to the body. We still have a good representation, but so much less, right? So our eyes, nose, face, upper lip, they take a huge area. And by the way, we're coming away from this thought process of homunculus. There are good studies that are now projecting us toward believing that this is not necessarily 100% true, okay? But this would be your sensory homunculus. That's the guy that feels. That's the humanoid. That is the representation of our brain. All right, now let's talk about the brain management. All right, so obviously different areas of the brain receive a lot of information, but but between birth to 5 years old, that's when the brain size increases by quite a margin. And also the complexity of connections increases a lot, okay? So, and this happens because of higher order brain development. That's why children, around that age suddenly start, you can teach them to read, et cetera, okay? Higher order brain function develops very, very rapidly. And higher order brain functions, we call them association areas, because what these areas do is that they take an information from one very specific area, take information from another very specific area, and bring them together. So you can make a judgment, and you can make a decision that best benefits you as you know it. So association areas in the brain integrate and process that integrated information from various areas: sensation, motion, history, and so on. For example, the ball is flying toward you. You maybe can hear the sound. Okay. Now, while you see that brain, let's say you see the ball. Okay, you see the ball. Maybe you hear someone yelling, "Hey, the ball is flying toward you, duck." So maybe you have a history of having been exposed to a ball flying toward you. So remember it, and then maybe you're already very well trained, then you are going to quickly catch the ball. Maybe not, and then you're going to duck. Okay, and maybe nothing works, and then you're going to get hit. That will be your memory, and then next time you're going to duck. Okay, so basically, this is where our cognition resides. Okay, the association areas. There is an association area called prefrontal cortex. Prefrontal cortex, PRE, is associated with PER, the personality. Prefrontal cortex is an association area that is responsible for different kinds of memory and your cognitive ability. You plan, you think in abstract ways, essentially what we are as a personality, okay? What's really interesting is that this area does not develop very early on, and that its axon myelination, so myelination of those axons, as I showed you earlier, actually goes into late 20s. So this is the area responsible for, who we are, personality, social behavior, etc. In addition to this, and this is a higher level of thinking, right? We have the more rudimentary ability to just feel the presence. Are we aware of things going on around us or not? So something that tells us that, hey, you know what, it's quiet now, sleeping is okay. Versus, that bee has been buzzing around me. What if I happen to have, what if I have allergy or something? I'm going to be very cautious. I'm not going to sleep. As soon as I hear that bee buzz, I'm going to wake up, right? Or alarm. You're sleeping very tight. You hear that alarm. So consciousness and alertness. What is responsible for this is the area in the brain called reticular formation, and part of it called reticular. Reticular means like a net, like a net, like a, essentially like a net-like structure, reticular, and activating system. So it activates this, it kind of like provides that level of arousal 24/7, where you know what's happening around you. Hopefully you do, right? Patients may lose it after severe accidents and so on. And so the way this reticular activating system, reticular formation, is structured is that we have a few nuclei, and I told you that, and I want to bring it home in the next slide, that actually a few slides down the road, that nuclei are the gray matter within the white matter in the brain. Actually, a few slides down when we talk about the basal ganglia. All right, so these areas of brain matter in the brain stem are interconnected. They have interconnected nuclei, and that's why we call it a reticular activating system, okay? So what happens here is that this area receives information, sensation. Is it hot? Is it cold? Is it loud? Is it safe? And so on. Processes this sensation, and then goes to the cortical areas and provides that information that the cortical area, the cerebral cortex, requires to make that conscious perception. And this area then makes that educated decision, if you will, okay? On the other side, after that decision has been made, many things kick into action. The same decision would result either in our muscles contracting. For example, if it's the bee, are you going to walk away? Are you going to run away? If it's a dinosaur, are you going to get very scared? If it's a tiger, are you going to get very scared? You're going to have that fight or flight response, right? Your heartbeat's going to go up, your blood pressure is going to go up, your eyes are going to open wide. Those are all automatic responses of the body, automatic or autonomic components. So motor and autonomic components. We'll do autonomic nervous system this week. So the brain has the right side and the left side, and the right and the left side are connected. So this is right, this is left. They are connected. All the white matter axons bundled together within the brain are called tracts. So they're like nerves, but within the central nervous system. So within the brain, within the spinal cord, we call them tracts, okay? So the brain has 3 tracts that actually cross from left to right. The most important one is the corpus callosum. Callosum means hard and tough, and it connects the left and the right cerebral hemispheres, half of the brains. So it crosses from left to right. There are also tracts, collection of neurons within the central nervous system, that do not cross, that run either on the right or on the left. Okay, those are called association tracts. Don't forget that association tracts does not mean association areas. Association areas are gray matter, which brings information together and makes those wise decisions based on history, based on knowledge, based on memory, and based on that information provided, right? So association tracts just move along the same part of the brain, same hemisphere. We also have projection. So CAP, cap the tracts, okay? Projection tracts leave the cerebral cortex and go down to the lower brain, the brainstem, and the spinal cord. So from the brain, they go down, and those are your projection tracts, and they project down. So projection tracts would go all the way here. And this, for example, is an association tract. How do I know? Why is it not a commissural tract? Because commissural tracts connect the left lobe here with that right lobe somewhere here. And there is no connection that we can see. Corpus callosum would be a commissural tract. This is an example called arcuate fasciculus. Arcuate fasciculus connects the Wernicke area responsible for understanding speech with Broca's area, which is responsible for making speech. And it's very important. It's a very important association tract. And you do need to know the speech processing. You need to understand that Wernicke area is responsible for understanding, and Broca's area is responsible for making speech. After Broca's area here has made the speech, it sends that information over here to the precentral gyrus, the premotor cortex, and says, okay, this is what you have to speak. Okay, so the motor Broca's area presents that what has to be essentially done, in what way the brain should move the lips, move the glottic area, and so on. The participation of the lungs are all controlled so we can talk to each other, speech formation. So the motor cortex is responsible for speech production. Broca's prepares that speech, a little bit of understanding. Wernicke is responsible for understanding. Prior to that, we sometimes need to hear information, right? And if you want to hear information, that information is heard here in the auditory cortex. That auditory cortex receives auditory information. That would be your superior olivary cortex. That would now tell, hey, you know, that's coming from the right. No, from the left. No, from above, and so on. All right. So auditory cortex is strongly associated with the superior olivary complex, which is responsible for sound localization and sits right here in the pons. So superior olivary complex gets you an idea of the direction. Okay, so now we're going to be talking. So we finished the cerebral cortex. Now let's talk about the limbic system. Okay, limbic system is very important because it is responsible for feeding, fleeing, feeling, fighting, and sex. We call them the 5 F's of our existence. Okay, so feeding and feeling, then you either run away or you fight, and obviously procreation. A part of the limbic system that sits right here looks like an almond, and so the Greeks call it amygdala, and that part, amygdala, is responsible for fear response, fight or flight. So patients who have damaged both of their amygdala, which is very, very rare, we have this information from the experiments. Those patients do not have any fear. They don't seem to recognize, their memory is also very bad. They don't seem to recognize the danger. So, you know, I may be, having a patient who has, sees a tiger and says, oh my goodness, what a lovely little thing. The fact that tiger may kill the patient does not occur to him. And we know this from the experiments on monkeys, where destruction of amygdala results in what is called Kluver-Bucy syndrome, hyperorality, hypersexuality, but also that lack of fear, as well as a loss of memory. So in general, limbic system has quite a few other parts as well, but a very important portion of the limbic system is called hippocampus. Hippo means a horse, campus means a sea monster. So this is the little sea monster, and this is the hippocampus. And hippocampus is obviously right here. This is your hippocampus. This is your hippocampus. So just behind the amygdala, okay, the green. And hippocampus is very important for memory formation. A lesion in the hippocampus will prevent a patient from forming long-term memory, and memory will not be consolidated. It's a part of the limbic system that is very, very important for storing memories, especially long-term memories. All right. In general, remember, limbic system, memory storage, limbic system, motivation. What is motivation? Motivation is emotion. Memory and emotion. Memory and emotion, limbic system. Limbic system. So this also includes kind of an interpretation of that emotion. And this interpretation is sent to the prefrontal cortex, right in front of the brain. And that's an association area which processes all that information, looks back into our history, our memories, makes the connection, and finally expresses our emotion. Okay, so the limbic system is composed of multiple areas that are responsible for emotion. If a patient has a problem with mood, we suspect either the limbic system or the frontal lobe. And once again, let's look at the prefrontal cortex right here, okay? This prefrontal cortex is the association area for personality, right? I mean, it develops up to 20 years of, after 20 years of age, that myelination I mentioned, that's your association area for the limbic area, limbic system, okay? All right, so memory. Regarding memory, you do need to know that sleep is very important for studying. Memory, we have sleep types called REM and non-REM. REM stands for rapid eye movement. During this rapid eye movement, patients' essential presentation, for example, oxygen consumption, a wave, brain waves recorded by the EEG, right? Electroencephalogram. That's how you record it. You put a bunch of electrodes around the patient's head, and you can record those mini currents. Okay, so during the REM sleep, which is very important for processing memory, our mind seems to be moving exactly as if we are awake. Very interesting, okay? And this, for example, is the deepest sleep. Look at the number of oscillations, the frequency versus this, almost none, okay? And everything in between, this is, for example, eyes closed. And this is already sleeping, non-REM sleep. All right, now let's talk. So just remember that memory and sleep, rapid eye movement equals to, has a role in memory processing, its consolidation. Okay, now let's talk about basal ganglia that I mentioned earlier. Basal ganglia are also known as brain nuclei. Most of the time clinicians use basal ganglia. Non-clinicians do mention brain nuclei, although it's not like clinicians don't know what nuclei is. So brain nuclei that you need to know are the lentiform nucleus, which is made up of the putamen here, and globus pallidus, which has two parts. It's both of those, okay? Globus pallidus, the pale body. There are many more, as you can see, subthalamic nuclei, amygdala, and so on. Okay, those are all basal nuclei, but substantia nigra, and so on. But what you wanna know is that those basal ganglia are collections of the nerve cell bodies. just like the gray matter, just like the gray matter here. This is also all gray matter. I mean, here it's like thalamus, for example, painted orange, but it's obviously not orange. It's got those black, darker gray areas. And so we're talking about the same like gray matter, okay? Same type of, same parts of the cell, non-myelinated areas of the nerves, of the neurons, no myelin, no masses of myelin. Masses of myelin equal to white matter, the tracts. Okay, so nuclei are clusters of gray matter within white matter. So just islands of gray matter within I. So normally gray matter is around that white matter, right? That's where all the axons are coming from. But sometimes we have that gray matter smack in the middle. Those are nuclei. Do not confuse this, the cell nuclei. Cell nuclei are obviously just the collection of neurons. What am I saying? That was me drinking water. Cell nuclei are obviously just that part, right? The nucleus of the cell, not your, the portion of the cell, a part of the cell, right? The command and control center of the cell. That's your cell nucleus. Now, brain nuclei are collections of many neurons. So we have nuclei, but that's not the point, okay? Brain nuclei are collections of neurons. Now, I already went over the lentiform nucleus made up of putamen and globus pallidus. Look it up on the picture. It is a very important site of control for subconscious muscular movement. For example, a portion of it called globus pallidus talks to the thalamic nuclei, thalamus, and controls the muscle tone. Okay, so your tone, are you gonna be very rigid or not, so that you can actually control your muscle movement in a without having to pay attention to it. Okay. Now there is another nucleus called caudate nucleus. There is actually there is a structure which we call, but never mind. There is actually a structure which unites both of these. It's called corpus striatum. It's not on your curriculum, but caudate nucleus. Cauda means a tail, and that tail-like structure is a part of the network coordinating muscles to produce a particular motion. For example, when walking, caudate is very important. When particular movement, like learning a dance, etc., pattern of motion, caudate nucleus becomes very important. Damage to caudate nucleus, you're talking about a disease like Huntington's disease, for example, where patients have dance-like movement, chorea, dance-like movement of the body, of the face. Well, face actually very grotesque movements of the face. Patients are miserable because those movements are all involuntary. So patients do not have a control of their facial muscles, don't have a control of any of their muscles. Horrible disease, a horrible genetic disease, Huntington's disease, caudate nucleus. All right, once again, this looks like a tail. That's why the caudate nucleus. Okay, and the white ball is the amygdala. There is your putamen, and the globus pallidus is that pyramid-like three-dimensional structure seen here. All right, so now let's move to diencephalon. So we did the cerebral hemispheres, we did the cortex, we did the limbic system and the basal ganglia. Now let's do the diencephalon. Diencephalon is pretty straightforward. There are three parts to it. Just remember, thalamus is in the word, thalamus is in the word, thalamus is in the word. So epi means what? On top, hypo means underneath. Epithalamus, hypothalamus. Three parts of the diencephalon, three main parts of the diencephalon. Let's do it. Start with hypothalamus. Hypothalamus is a portion of the endocrine system which we'll be studying, and one of the actions that it involved in intimately is the sleep-wake cycle, sex hormone production, and so on is another one, okay? So hypothalamus connects the nervous system to the endocrine system, okay? Now, next portion would be thalamus. And let me, at this point, let me remind you of RAS. RAS, the reticular activating system, which is a part of the reticular formation. I told you that it provides that ability that we are kind of 24/7 watching our environment. We're aware of our surroundings, right? Well, that's the reticular formation. What thalamus does, it enhances it even more, and it serves as a relay point for sensory information, and it projects to the primary sensor region, so the post-central gyrus, that's where the sensory homunculus is. But what it does is it filters out noise, irrelevant noise. For example, I'm yelling, I'm giving out, for example, there are 100 of students sitting there, and I'm telling them, question number 1, this is what you're going to see on the exam. Question number 2, this is what you're going to see. And then let's say there's a loud music playing in the background. I don't know, some popular superstar singing. I know that most of you are going to be listening to me because I'm giving you questions, right? So no one will care about the music at that point of time. Now, let's switch the situation where I'm yelling the questions out at you after the exam time, and you have like a really nice music. So what's going to happen is now you're going to concentrate on music. So if I'm a coach, I'm yelling out orders, or suggestions. You're going to hear me with all of that background noise, right? So that's thanks to the thalamus, okay? Epithalamus is the roof of the third ventricle, roof of the diencephalon, and it's the posterior most portion as well. And posterior portion of the epithalamus houses its largest portion, and that's called a pineal gland, also known as epiphysis. Epiphysis, okay, pineal gland, different words, different names, and the stalk is actually contains the habenular nucleus responsible for pain, but the pineal gland actually secretes melatonin. That's a hormone that some actually use as a pill to enable you to sleep. So you want to say, all right, this is nighttime. I want to sleep, not day. This is the night part of the sleep-wake cycle. So if we think of the day versus night, circadian is the word that means around the day. Circa is around, dia is the day, circadian. So pineal gland secretes melatonin, which regulates our circadian rhythm. When you take melatonin, you become drowsy, and that helps you to establish that day-night rhythm. All right, at this point, let's talk about the next portion, which is not really directly important, but is often mentioned in your curriculum, and that's the brain stem. Brain stem is the lower part of the brain. See it here, all in green, and it contains 3 parts: the midbrain, the pons, and the medulla. The midbrain, the pons, and the medulla. Okay, what it does, it connects the cerebrum with the spinal cord, okay? And therefore also controls many basic functions. We'll see what they are. Brainstem, midbrain, pons, medulla. Please remember, by the way, this is your pineal gland. If you look from the back, it's above the these 4 structures, 1, 2, 3, 4 quadri. quad. Okay, we'll go over that in a few. That are a portion of the tectum. Okay, so a pineal gland produces melatonin. Now, yeah. So once again, a midbrain. Okay, mesencephalon. So this is the portion of the midbrain called tegmentum right here. And this is the portion that looks like a roof. That's your tectum. This is the tectum from the back. So I'm looking at the brain like so. Okay, tectum. Tectum means the roof. Okay, we'll go over it in a second. I'm strengthening, I'm mentioning it because there will be a term coming up. Okay, and that term is called quadrigemina, corpora quadrigemina. So this tectile plate, okay, plate. You know what a plate is, right? Like a plate, like a roof-like structure here in this case, okay? Contains 4 of these structures. We call them together corpora quadrigemina. 2 of them are on top, so we call them superior. Superior. We call them superior colliculi. Okay. 2 of them are at the bottom. We call them inferior. These are the inferior ones. Okay. So superior colliculi are responsible for vision because they're responsible for visual processing and eye movement. Okay. Inferior colliculi are part of the auditory information processing machinery. Inferior colliculi, superior colliculi, known together as tectile plate or corpora quadrigemina. Here is your plate from here, from the back, okay? Here is the plate if you were to cut out the cerebrum. Please remember, we also have a portion in the ventral. So this is from the back. This is from the front. From the front, you can say anterior or ventral. So the portion of the midbrain that is in front is called tegmentum. Tectum is behind it. In between is the cerebral aqueduct, okay? And this tegmental area has an area which produces dopamine. And that area is called substantia nigra and ventral tegmental area. Substantia nigra is damaged in patients with Parkinson's disease. So it's missing all those neurons in the tegmental area, and patients have a problem with motion. Motor control is damaged, okay? Because this area, the midbrain, is responsible for motor control and to some extent also sensory processing. If you think about the corpora quadrigemina, this plate that is responsible for vision, superior colliculi, and audition, the inferior colliculi. Okay, so in addition to that, if you were to look from rostral, from anterior, from ventral, you can use any of these, right? You're gonna see that slicing this midbrain like so gives us a cut like the one here. So this is your cerebral aqueduct. Above it are the 4 colliculi, 4 colliculi, because they are a part of the tectum. So superior colliculi for vision, inferior would be below that for sound, right? This is the cerebral aqueduct. Remember, that's the hole through which the 3rd ventricle communicates with the 4th ventricle, okay? Now, look at these 2 structures. They look like feet, and these are called cerebral peduncles, cerebral, not cerebellar, cerebral peduncles, because this is where the motor tracts, so projections of axons from the gray matter in the prefrontal cortex from the gray matter nerve cell bodies originate the axons, right, that are wrapped with myelin. So these axons bind each other, pull together, form wide nerve-like structures in the central nervous system. We call them tracts. These tracts go down to the rest of the body, okay? And while they're crossing the area of the midbrain, we call them cerebral peduncles because they look like little feet and they're on the anterolateral side of the midbrain, okay? So they're present on the ventral aspect of the midbrain and they contain these descending axon bundles. They go down, down. This is the control of our biceps, of our triceps. So this is all voluntary. You want to flex your biceps? Sure, go ahead. You want to flex your triceps? All right. You want to walk? All right. That's what these tracts are for. Do not confuse it with the middle cerebellar peduncles or superior cerebellar peduncles or inferior cerebellar peduncles. For example, the middle cerebellar peduncle connects the cerebellum to the pons. It's not the same thing, okay? This is your cerebellum, the pons is in front of it, so that's a connection that is different than from cortex, the gray matter, to the spinal cord, okay? Okay, so at this point, let's talk about so we did the mesencephalon, now let's talk about the metencephalon, okay? Metencephalon contains the pons and it contains the cerebellum, okay? So a significant portion, so we're coming to an end, let's focus. I also need to focus, that's been a lot of talking, 2 hours almost, but not quite yet. All right, so a significant component of the brain that we can clearly identify under the cerebrum is called cerebellum, the small brain. You can see it right here. You can see it right there. That's the portion with kind of a tree-like structure, right? And that tree-like pattern is called arbor vitae, the white matter of the cerebellum. Everything around it is the gray matter, the neurons, not the axons. White matter are the axons. It's very distinctive, so it's all often mentioned, okay? So just a reminder again, middle cerebellar peduncle, middle cerebellar peduncle. So the cerebellum has to hold on to the pons, right? It essentially makes the pons by reaching out and grabbing and holding around. And that's your middle cerebellar peduncle. Middle cerebellar peduncle hugs the brainstem, okay? Hugs the brainstem. All right. Now, what is the function of cerebellum? So I'll explain to you in 2 words. How does a patient who has a known alcohol problem history present? Kind of shaky, right? Kind of shaky. That's a damage to the cerebellum. Damaged cerebellum happens in a loss of smooth moves. So what the cerebellum does, it receives the information from all of our muscles, okay? And relays it, cerebellum, and relays it to the primary motor cortex. That's the frontal area right in front of the central sulcus, right? So that's the frontal area, for example, that moves the mouth. That's also the frontal area that moves the arms, the legs, and etc. in a smooth way. So a patient who has a damaged cerebellum, tell them to close their eyes and just do a finger to nose test so that they make a large circle and then try to touch their nose. They're going to be like this, and they usually overshoot, okay? So that's the problem with cerebellar damage because it's responsible for coordination. So it adjusts and ensures that the motion is smooth by tweaking the motion of the muscle. So it says to the arm, hey, don't move too far away. That's the target. That's why You know, gymnasts, for example, have a very well developed coordination. So obviously very well developed cerebellar connectivity, right? So what it does, it tells the cerebrum everything about the body. It provides an information. If you raise your arm, you know that your arm is raised even when you close your eyes, right? Cerebellum conveys that information. That information is called proprioceptive. So it's the information that is our own. So where are you in space? Okay, so proprioceptive information from the muscles carrying a voluntary, very important, voluntary information to the primary motor cortex. You should be familiar with that term, primary motor cortex. It's part of the frontal lobe that I showed you earlier, not the postcentral gyrus. That's the precentral gyrus. That's your motor homunculus, okay? So mesencephalon is your midbrain, okay? Now let's talk about the myelencephalon. That's your medulla. Medulla is just the area in the brain that is the lowermost portion of the brain stem. You know, the midbrain, the pons, and the medulla are three parts. And what it does is that it has other nuclei that are very important. So what medulla does is that it controls the vasomotor activity. What does that mean? Contractions and relaxations of the blood vessels, vasovasculature, motor. So it tells the blood vessel, please constrict or please relax. Those are all orders coming from medulla oblongata. Okay. It also says the same thing to the heart, because heart is very closely interrelated to and interconnected to the blood vessels, right? So it controls the heart rate and how fast the heart beats, but also how strongly it beats. That's very important. Okay, you're going to study the difference during your cardiac classes. One very important thing that's never to be forgotten about medulla is that it controls our respiration. It says how fast or how slow you should breathe or breathe at all. So, for example, if there is a damage to the brain, some of it may herniate, squeeze through holes of the brain, and compress the medulla right here. What will happen is that the respiratory center will get squeezed, patient will stop breathing. Very dangerous, because medulla performs vital functions: heart, blood vessels, breathing, heart, blood vessels, breathing. One more thing that medulla has that is pretty unique, that's a crossing. A crossing in Greek is called decussation, Latin actually, decussation. So there is a crossing. This happens at the level of the medulla, so we call it medullary decussation. And this is the crossing of the tracts that we saw earlier, the peduncles of the midbrain. Okay, so those midbrain peduncles go down, and the tract itself is called pyramidal tract, because look at those pyramids. Do you see those? They kind of look like so. So the Greeks looked at them maybe and thought, you know what, that looks like a pyramid to me, and I'll call it pyramidal decussation. So here are your medullary pyramids, right? This is all tracts coming down, passing that cerebral peduncle, still going down. This level, it's a pyramid. Now look here, this level, this pyramid goes to the side, this pyramid goes to this side. And that's why when we move our right arm, for example, actually 90% of the brain neurons are on the left. So the left controls the right, 90%, right controls the left, about 90%. All right, and that's called pyramidal decussation. That's the crossing point of axons for the corticospinal tract. And remember, this is all voluntary. Corticospinal tract is when you tell your muscles of the biceps to contract and so on, okay? So That was a super long class, 2 hours. Anna, was that a review or let me stop?